Jenna.
And I'm Steve.
Welcome to another Field Trip Friday. Where are we today, Steve?
We're at the Nicholas School of the Environment at Duke University, and we're going to be meeting Melissa Chernick and Lindsay Jaspers, who work in the DiGiulio lab, which studies fish.
Whoa.
Yeah, they study fish that have been adapted to particularly polluted river in Virginia called the Elizabeth River.
Yeah, I'm familiar.
You're from Virginia.
I am from Virginia.
We're going to learn a bunch of cool stuff about how they approach scientific problems.
I can't wait for that.
It's going to be super fun.
Let's go take a look.
Let's go.
My name is Lindsay Jaspers, and I am a postdoc in Rich DiGiulio's lab in the Nicholas School of the Environment at Duke and part of the Duke Superfund Research Center.
I'm Melissa Chernick, and I'm a research technician in Richard DiGiulio's lab as well as David Hinton's lab, both of which are in the Superfund Center at Duke University.
Lindsay, Melissa, thank you so much for allowing us to spend some time in your lab today.
Sure.
Can you tell us a little bit about what you all do here at the Nicholas School? We study populations of fish. So we have one fish that we work with is the Atlantic killifish that lives all throughout the East Coast. But one of the places where we're primarily interested in studying these fish is the Elizabeth River, which is a very polluted place in Virginia. It has decades of pollution, and there's fish that have adapted to that pollution, and they've been able to withstand that and live there successfully for decades and decades. And we're really interested in studying how those fish have been able to survive there and adapt to that pollution. So we study different aspects of how they've adapted, what sort of costs there are to being adapted. So are they more susceptible to other stressors because they've adapted to this pollution? And then we also study fish like zebrafish, which are just lab species where we can have different strains that allow us to test all different types of proteins and responses to chemicals, and we have a lot of methods that have been specialized for zebrafish that we have enabled us to test a variety of different things with those fish. Maybe you can add some more to that.
Like Lindsay said, we study Atlantic killifish. We go up to the Elizabeth River in Virginia to collect the killifish every summer. We bring them back to our lab, and we have a nice tank setup, aquarium setup for them to live in, and we will collect their eggs, their offspring, in order to do some very controlled laboratory testing. These sites were designated as Superfund sites by the US EPA.
And so that's Superfund.
Superfund with a D, yes. Superfund with a D. They're not super. We have fun when we go to these sites, but I wouldn't classify them as super fun.
Right.
Yes, but super fun. And what this is it was a measure that was put in place decades ago where industries that cause a lot of pollution put money into this super fund in order to study effects of that pollution and clean up those messes. And so it's a pool of money that the EPA or other agencies can pull from in order to clean up these sites.
So yeah, what is some of the benchtop stuff that you find yourself doing? Like probably prepping like various solutions and things that you use for your
Right, so
fermentation.
we have these are sort of, we have a big cabinet of chemicals, but these are a lot of our commonly used chemicals, and we have pipettes and instruments that we need. And then we have pH meter, a microbalance, which is a very sensitive scale, and what's called a dissecting stereo microscope.
Nice.
So we can put whole fish under here for measurements. So for example, we have a pair of calipers here if we want to measure fish length or girth, then we can do that. But any sort of preparation is done in this area. So when we do embryo exposures and we want to check on them every day to see how they're doing, how they're developing, then we will put them under this stereo dissecting microscope here. And we have a camera on the microscope, and it's attached to this computer here where we can take pictures of those fish.
Cool.
Very cool.
Oh, it's so cool to see that
There we
blood
go.
flow.
Okay.
Yeah.
Yeah. So the eyes, the head, so he's wrapped around the back of the yolk, and this is his tail coming up right here. And then this is the heart, so the atrium and the ventricle. These fish have 2 chambered hearts instead of 4 like we have. So down here the blood is coming through what are called the yolk veins to collect in the sort of this confluence. It's called the sinus venosus. And then it goes into the atrium, the ventricle, back through the body, and it loops around again.
Cool.
And so this is the egg envelope, or the chorion. That's the protective layer of the fish. Down here is the yolk. These little bubbles here are lipid or fat droplets, which the embryo, especially right after it's hatched, when it becomes a larvae, will use as an energy source for those first few days before it can catch its own food. So it'll be on the yolk sac about 3 days. Depends on how active the individual is and how they are using up their energy. But you sort of have a head-on view. He's looking out at you in this picture. And so his mouth is right here. And once the yolk is gone, some of this stuff will sort of migrate back into the abdomen and then develop a little bit more. So the liver is there. It will develop more into a mature liver. Same thing with the heart. But for all intents and purposes, he has all of the organs, at least in very early developmental stages, that he'll have as an adult here. So these fish are what we sort of sometimes call as stay-at-home fish. If you like that, especially in these times, we know what stay-at-home means. And they pretty much stay in the same area for their entire lives. So where they are hatched is where they live. They only move about 30 or 40 meters is their home range, which means if you have a high level of pollution at a very specific area, the fish are there. They're not going to move out to a cleaner area. It's just not in their life history or their evolution to do that. And so you either adapt or you don't survive. And so it's what we call a very high selection pressure on the population. And that has caused these fish to adapt to these very highly polluted environments. And so we go and we collect fish from these populations at these very specific sites from these different companies. We bring them back to the lab and we try to figure out how they do that. So this is the sump tank or the water reservoir, and it moves up through these pipes here to the 2 test tanks. And the fish are in these chambers within the test tanks, and the tanks have pumps, temperature sensors, and oxygen sensors in them. So the water is coming from this water reservoir. You can see it pouring in here. It's coming through here and then back down to the reservoir. So it's constantly cycling in here. So we have fresh water that's oxygenated going through to the tanks here. And then within the tube, at the beginning here, you can see sort of the gray honeycomb-like piece. And that is a fan that we can turn to start moving the water if those are the tests we want to do. And then all of the cables go back here to the computer which is taking the readings from those sensors.
Cool.
Right now, resting metabolic rate or oxygen consumption is what's being measured. But if we were to turn on the fans, we could take a lot more measurements.
It's
So things
basically
like the
they're
velocity,
standing on a treadmill but not moving.
right? Sort of like the stamina, or how long they can keep swimming before they get tired. That sort of stuff are more active metabolics. So these fish were exposed to flame retardants when they were embryos, but then they were grown up in clean conditions. So these are the equivalent of about 65-year-old humans. So,
Cool.
So what we try to do, and this is where collaborations with multiple labs as well within our Superfund research program or center comes in, is in those lines of evidence we look at what we call different levels of biological organization. So the whole animal to an organ, to its tissue, to a cell, to genes. And if you look at these different levels and you find the connections or links between them, a full story starts to emerge and you start to get closer to solving that puzzle.
Yeah, really cool. So we got to see several of your really cool spaces and some of the amazing tools that you use to kind of probe these questions. And it seems like it's really important that this is like, not only do these fish adapt, but so does your lab. It's kind of like evolved and grown over time. New postdocs come in, new graduate students come in, they have their own projects, they have their own funding, you know, all sorts of things kind of affect where the storyline goes.
One good science question spawns 10 more science questions.
Right.
And so we always have something to answer in the lab.
Yeah, once we answered one question, it just created like 10 more questions that we could answer. So once we figure out, oh, well, these fish have adapted to pollution, well, how have they done that? And then we can ask, okay, well, now we've remediated these sites, how do they respond to that? And do they go back? And what are the costs and the trade-offs of adaptation? So once we're answering one thing, it just builds upon more and more questions.
And in the laboratory, we design experiments where typically we change one thing. Everything is the same, we have different populations, but we change one thing. We add our chemical mixture. But the environment is a lot more complicated and complex. So studies like Lindsay's, that's looking at temperature, add in another thing. So you have all sorts of stressors in your environment, temperature, as she said before, low oxygen, and when we look at things in combination, the questions become that much more complex. And so those will spawn even more questions in the laboratory. So it certainly keeps us busy.
Yeah, absolutely. It seems like the journey is never over.
It's never, and it's never over.
Lindsay, Melissa, thank you so much for letting us join you today at your lab.
No problem.
No problem.
Thanks for coming.
No problem back at you.
Yeah, thank you so much. It was awesome to hang out with y'all. And for those of you watching, we're going to have a live Q&A with Melissa and Lindsay right after this video. So stay tuned for the live Q&A. We'll see you there.
See you there.
Bye, everyone.